Great. Thank you so much for this wonderful opportunity to share with you some of the data from the trial that Dr. Mohamedy presented earlier. He did kind of steal some of my thunder, but I'll try to go quickly through the parts that he showed and maybe go into a little more about what conclusions we can glean from the data that we've generated so far. So he had mentioned this as well. One of the things that has changed oncologic practice, I'm a medical oncologist, medical oncology fellow, in many cancers has been immune therapy, especially checkpoint inhibitors, where in many cancers, as you probably know, like melanoma or lung cancer, the introduction of checkpoint inhibition has led to subsets of patients with very prolonged or durable responses to therapy. However, glioblastoma doesn't seem to be one of these diseases, and really a number of the larger phase 3 trials using checkpoint inhibitors, both in the primary setting and the recurrent setting, have failed to show a consistent benefit in overall survival. So there's really an unmet need here to develop strategies that overcome checkpoint inhibitor resistance in GBM. So why is GBM so resistant to immune therapy? One of the main reasons for this is that up to 50% of the tumor mass can actually be infiltrated by immune suppressive cells, especially macrophages and microglia. As you can see in the bottom left there, it's a human GBM specimen that's just massively infiltrated by these myeloid cells that express IBA1, CE204, many of which are going to be suppressive to the immune system in the tumor. And just to convince you about how critical these cells are to inhibiting the immune response, on the right side there are Kaplan-Meier curves from a mouse model. So this is a genetic mouse model of spontaneous glioblastoma using the genetic deletion of mutation of PDGF. And in these mice, if we deplete macrophages, either with drugs or genetically, most of the mice are actually able to control the formation of tumors just with their immune system. So these mice develop tumors, but then the tumors don't grow beyond a certain point and the mice survive. So as we think about how do we find new and better targets for immune targeting strategies, we need to start from understanding the biology of the tumor microenvironment. And the question that Dr. Mohamedy had alluded to is, are we studying the correct region of GBM? So, usually before we thought about sampling different regions, we look at GBM tumors and say, this is the tumor. And then, a neurosurgeon goes in, takes out a piece from somewhere. It eventually comes to the lab and we try to identify therapeutic targets. And really, many studies used to identify these targets, again, treat GBM as a homogeneous tumor. So we went back and there are different ways to divide up a tumor. The way we did it was using imaging, using MRI technology, and divided the tumor into the core, MRI enhancing rim, and the non-enhancing rim. We saw that earlier. And just to reiterate, we don't really know a lot about the distinct biology of the different regions. And really, we thought that understanding how that non-enhancing rim is different could lead to development of better strategies for GBM. And just to remind you of a study that I'm sure you're all familiar with out of UCSF, this is one example showing that patients with wild-type advanced glioma or glioblastomas, if you're actually able to resect the majority of the non-enhancing rim in the blue there, you end up getting better survival than you do if you're not able to resect the non-enhancing rim. And I know this is an area of ongoing debate in the field, but in any case, there is indication that the more you're able to remove of the non-enhancing rim, you're affecting disease outcome. So before I get into this, so basically that suggests that region is important for recurrence of the disease. So again, we also use the Myriad device to sample different regions. We saw that earlier, so I'm not going to repeat it. And the hypothesis for the study I'm going to show now is that the non-enhancing GBM rim is going to be characterized by a distinct immune microenvironment. So we thought, it's the enhancing, there's edema there, there's inflammation, so it's going to be very different from the non-enhancing, where you likely have a lot of interaction with neural cells, right? Neurons and glial cells. And we were surprised that was not going to be the case, and I'll show you in a bit. So the experimental approach, you saw it earlier. We used the MRI to map 3 different sampling sites. All of this, we have data, so we can go back and actually combine our data with radiomics, which we have not done yet, but are planning to. And this is just a schematic of the workflow where we quickly get the tissue to the lab. Some of it gets flash frozen, some of it gets digested, and the data I'll be showing today is from immediate flow cytometric analysis before the cells ever get frozen. And the reason for this is that some of the cell populations we study, so specifically granulocytes, granulocytic MDSCs, these are important suppressive cell populations, they actually get lost in the freeze-thaw cycles. And there are several papers from immunology studies showing that just by freeze-thawing, you're altering the representation of immune cells. So we wanted to immediately get that data and get an accurate representation of the immune microenvironment. So a little bit more about the patient characteristics. There was 20 patients with primary glioblastoma. You can see the ages there. More than half were over 65. There were 8 females in the study. 5 of the patients were MGMT methylated and 5 were EGFR mutated. We used 2 different panels, and all this hopefully will be published soon, but just to quickly show you the panels that we used. On the left was more of a general panel that would look at different major immune populations, including the macrophages and the suppressive myeloid-derived suppressor cells. And then on the right, we have a panel that looks at T cell phenotype, the activation status, exhaustion markers, and their memory phenotype. We saw a little bit about this before, but I'll go into a little more detail since I have time. There are several ways to look at viability, and we've done both immediately trypan blue exclusion. You basically put a dye on the cell suspension. Live cells don't take up the dye, and we saw that probably because of all the debris in the samples that come out of the tumors, that turned out not to be a very accurate way to be able to discriminate live from non-live cells. But when we used flow cytometry, we're able to much more accurately gate on the live cells. And as was alluded to earlier, the viability of the non-enhancing rim was higher. But overall, the viability of the samples was acceptable. It was over 80%. I think there's 2 ways we can explain the differences that we saw between the regions. One way is, could it be a technical artifact, right? So different areas could be more susceptible or less to death outside of the body once you have the tumor specimen. But honestly, I think part of it is just due to the biology of the different regions. So it would make sense that if you have a necrotic core, you probably wouldn't have as high viability as the non-enhancing rim, which is closer to the normal brain tissue, let's say. Again, we saw this earlier, the overall immune cell population was less in the non-enhancing rim, was less abundant, and this is out of all the live cells. And I think we haven't formally looked at this yet, but plan to do it in the next steps. There's probably going to be a higher representation of neural cells in that non-enhancing rim, so cells from the healthy brain, essentially, and understanding how those cells also change when they're in close proximity to the tumor is a whole nother field of neuro-oncology and interactions between neural cells and tumor. So this is the heat map that Dr. Mohammadi showed. But what it's trying to say is that overall, the different cell subsets that we looked at tend to be less abundant. So the pink there means less abundant in the non-enhancing rim. It wasn't one particular subset that was more affected or less. Nonetheless, if we do look at each one of these major subsets, so the one thing I'd like to draw your attention to are the different scale bars, and to look at how big of a variability there was between the tumor specimens that we obtained. And really what this goes to say, and something that we've known from other studies, is that there's really a huge variation in the biology of these tumors, even looking at a very superficial level of the representation of different immune cell subsets. And I think one way we can make use of this data moving forward is really to be able to expand the number of patients that we enroll in trials like these, so then we can actually go in and subsegment maybe different mutational statuses or different patient characteristics to group patients, and then make meaningful conclusions that can help guide further therapeutic development in the future. So let's focus a little more on the T cells, and I'm gonna again try to give some meaning to the data that Dr. Mohammadi briefly showed earlier in the day. So one of the things that we looked at was the ratio of CD8 to CD4 T cells. So as you probably know from your basic immunology, CD8 T cells tend to be the ones that are more capable of killing tumor cells, whereas CD4 cells maybe not so much. There are some nuances, but in a very superficial sense, it tends to hold true. And we saw that not only were there fewer overall T cells in that non-enhancing portion, but also the abundance of the ones most able to kill tumor, CD8 T cells, were lower. So that could suggest that in that region, the immune system is less able to recognize and kill any residual tumor cells that are invading the healthy brain. We looked at memory phenotype, and we saw similar pattern across both CD4 and CD8 T cells. Again, because of the patient variability and the relatively small number, it was statistically significant only in the case of CD4 cells. And here specifically, what we saw was a shrinking of this little yellow portion, which are the central memory cells. And these are the cells that tend to be more long-lived. A lot of the CAR T trials, for example, have shown that this is the population that's important to sustain in the body in order for the T cells to be able to fight, recognize, and kill tumor cells. So together with the CD8 data that I showed in the previous slide, again, this suggests that there could be a further impairment of the ability of T cells to kill cancer cells in that non-enhancing rim, which again suggests that we may need to tailor any immune therapies to that region and not use the same things that we do for bulk tumors. Okay, so all these are phenotypic markers of memory and different cell subsets. Are there any differences in T cell functional markers? So I'm just summarizing the data here. This was very surprising to me. So I expected that there would actually be differences in exhaustion markers and activation markers, and in fact we didn't see any consistent patterns. So regardless of which of these exhaustion or activation markers we looked at, we saw that their levels were very comparable across the different regions. So what this told me, the way I interpreted this, was that yes, there are changes in the overall abundance of different cell types and of immune cells in general, but the biology of that initial tumor does to some extent inform the remaining immune cells that are in the non-enhancing rim. So there is somewhat of a signature of activation and exhaustion marker that persists after the resection of the tumor. And again, so that data did give us some insight into the function of the cells, but it really was just a static picture of markers that are expressed. So what about a way to address their actual functional capacity? So we obviously can't do that in the person, but we can take the cells out and see in vitro how able they are to show functions that are required for killing tumor cells. And the way that we do this is we have our frozen samples. We can thaw the cells a lot of which are immune cells. We stimulate them with chemical stimulation. PMA is the name of the chemical. And then essentially 24 hours later, detect cells that are able to produce cytokines that are important for tumor cell killing. And again, I had expected there to be differences, and in fact there were not. And you know, one thing I want to highlight is all the analysis we do, we always analyze comparing the matched samples. Okay, so if there are patients that group high or low, and there were differences, we would still be able to detect it. And we were not able to. So granzyme B is one of the killing enzymes, TNF alpha, interferon gamma. These are important cytokines for tumor cell killing. So it didn't seem that there was really any functional difference between cells in the different regions. So again, this speaks to a retention of some kind of a functional signature even after resection in that non-enhancing rim, which means that the cells should still be able to kill cancer if they're activated in the appropriate way. So, you know, in the future and ongoing, actually, what we're doing is trying to accrue more patients in subsequent studies, so we can start to subcategorize different patient groups and see whether we can draw some more meaningful conclusions that'll help guide future therapy development. And also we're in the process of doing single cell RNA sequencing of both the tumor and the immune cells to really get down to the RNA level of how these cells are different in the different regions and how they talk with each other. So just to wrap up here, some conclusions. So that we've seen this from a few different talks now. You know, the NICO TPS system, Myriad system, has allowed us to get high viability samples from spatially registered regions of glioblastoma. Overall, we saw the non-enhancing rim had fewer immune cells and underrepresentation of several suppressive myeloid cell subsets. So there are differences in that respect. But on the other hand, the CD8 T cells, even though they were fewer, they retained their cytotoxic function and their activation markers, as was the case in the other regions. So, you know, we think that the non-enhancing rim is not immunologically cold, and it's just a matter of understanding better its unique biology in order to design better immune therapies, perhaps in the adjuvant setting. So I'd like to thank, you know, in addition to the people that Dr. Mohammadi mentioned, obviously our patients, my mentor, Justin Latty, and the lab, as well as the other people that helped, and of course, Nico for all their support and collaboration. Yeah, thanks.
Thank you, Dr. Watson. Great talk, great work. You sort of alluded to CD4 cells, and you just said they were sort of Can you be more specific, their role, or what are they doing there?
Of course. So yeah, I mean, CD4 T cells in the tumor microenvironment are well studied. You know, they're a big subset that's important in cancer are T regulatory cells that I didn't get into, but Tregs also were similar across the different regions. There are also, you know, they have an important quote unquote helper function. So they essentially help the other cells work. They provide cytokines for the CD8 T cells. And finally, there are so-called cytotoxic CD4 T cells that share a lot of function with the CD8 cells in their ability to kill tumor. But you know, with the superficial analysis that I was showing, it's easier to think about just CD8, CD4, because those other subsets I talked about, they need additional in-depth analysis.
Okay.
Great question.
What do you think about continuing the study with Dr. Aliwala? I think there's 120.
Yeah, absolutely. So yeah, I mean, we're all part of the same team. So that's the whole idea again is to really expand this cohort, because the major limitation, you know, having personally analyzed all of this data, was the huge variability. And I think the only way we can overcome that is by increasing the N number and thinking about smart ways to group patients for analysis.